Hi everyone, thanks for checking out this video. So this is going to be the first video in this new series I'm making on this course called Intro to Bioinformatics. And this is going to be a course that's going to be an introduction to bioinformatics for beginners, people who are like new to this field or possibly just curious about it and want to learn about it. And this course is going to assume a little bit of a programming background. So we're going to be using Python. So it'll assume some prior programming knowledge, but it doesn't assume any prior biology knowledge. So if you're a total beginner in biology and you don't know any, and you don't have any biology background, that's okay because we'll cover all of the relevant biology. It'll be assuming no prior knowledge on that. And yeah, it's going to be like a quick video just introducing the course before we get into the material, just kind of giving like an overview. So to start, I figured I'd introduce myself quickly. So my name's Mike. I currently work as a research scientist at the University of Pennsylvania in Philadelphia. Yeah, my job there is mostly bioinformatics, a lot of data analysis, a little bit of machine learning, mostly applied to circadian rhythms in gene expression. It's pretty cool. Basically, I feel like I am getting paid to do my hobby for a job, just working on cool research like professionally. I really like it, and it's pretty fun. Before that, I completed a PhD in bioinformatics at the University of Delaware, and my dissertation there was mostly on like mathematical biology and models of randomness in cell biology. Also a little bit of like transcriptomic data analysis. If you guys don't know what that word transcriptomic means, don't worry because we'll talk soon about what that means and what kind of data analysis that is actually. And yeah, before that, I had like a lab assistant bioinformatics job working for Clemson University, although I actually worked remotely, so I didn't actually live at Clemson. I worked remotely for this lab at Clemson, basically just doing some like Python programming work for them. And before that, skipping over a couple things, my undergrad education was actually in something pretty different. I studied economics for my undergrad degree, and I minored in math and computer science. So it was a lot of like quantitative work, a lot of programming, a lot of like data analysis and math, but no biology at all. I didn't take any biology classes in my undergrad, and really didn't take any natural science classes in my undergrad at all. And so the reason I'm making that point to you guys is just to make the point that even if you don't have a biology background, if you have like a quantitative and a programming background, it is possible to switch into this field. And so that's what I did. When I got this first bioinformatics job, I really only had a programming background and like a quantitative background. I really didn't know any biology, but I was able to get my foot in the door with the programming knowledge I had. And yeah, just want to make the point to you guys that that is possible to do. So if you're watching this video and starting the series with no biology background, but you think you might be interested in this field, it is possible to switch into this field coming from only a programming and like quantitative background, even without a biology background. So yeah, just want to make that point to you guys. Okay, so what is bioinformatics? In very general terms, it's basically a field that's a combination of biology and computer science. And there's also some other fields that are like partial synonyms that it's kind of related to. So you guys might hear all these terms like computational biology, quantitative, systems, theoretical, mathematical biology, biostatistics. All of these fields are like partial synonyms. They're pretty related to each other with a lot of overlap. And what they all have in common and what they all share with bioinformatics more broadly is that they're all like subfields of biology that don't involve doing experiments and are more about like quantitative work, quantitative analysis, analyzing data, like math modeling, statistics, things like that. So all of these fields, I think of them as kind of being under the same umbrella at least, and all being like pretty related to each other. And I think of the big difference as being between experimental biology, also called wet lab biology, and like computational biology, also called dry lab biology. So that's where the big difference is. So I think like I could pretty easily like switch to working in any one of these subfields, these quantitative subfields, but I could not very easily switch to doing experimental work, because if I did that, I would be like a total beginner at that. I really don't have any background in it. So I think of that as being the big difference between like the experimental and computational biology. But I think of all these things as being partly synonyms and all kind of under the same umbrella that bioinformatics is also part of. And within this like broad category of like quantitative computational biology, there's a lot of topics that are interesting that we won't be covering in this course. So one of them is math modeling of biological systems. But if you're interested in this, I do have some other videos about it. So check out my other videos if you want to learn about this. Algorithm design for sequence processing. So a lot of computer science work related to that. But yeah, we won't be touching on that in this series. Low-level bioinformatics, that's basically like processing the raw output of sequencing experiments to get it into a more like recognizable like Excel file type format. Yeah, pretty important, but we won't be really doing that in this course. Software development, biomedical engineering, public health and clinical informatics. Again, all of these things pretty interesting and cool fields of research. But yeah, we won't really be touching on them in this course. So our focus for this course is going to be data analysis for biology. Basically talking about some types of common data sets in biology and how to analyze them. And yes, I picked this topic, one, because it's a little bit easier than some of those other things, a little bit easier than kind of like the math modeling and like the sequence analysis type stuff. It's a little bit easier for beginners. And also, this is probably the most direct way to get your foot in the door if you're trying to switch into this field professionally. This is like the quickest way to learn a skill in bioinformatics that will make you actually useful and employable to a biologist. So if you learn how to do this, learn how to like work with some of the common data sets, learn how to do some like basic data analysis and visualization, that's the quickest way to make yourself employable and useful to a experimental biologist. So if you're trying to get your foot in the door in this career field, then this is probably the most direct way to do that, I think. And I guess like the third reason is just that it's actually like interesting and fun to do this type of work, basically. And also potentially useful for whatever biology topic you happen to be interested in. A lot of what we're going to be talking about in this course is very general, in the sense that the techniques we're going to be talking about could be applied to very many different biology topics, whether you're interested in like cancer or biology of aging, or fundamental biology, or like plant biology. Really, for many different topics, the techniques we're talking about are going to be general enough that you'll be able to apply them across like a broad range of possible topics in biology that you might be interested in. And yeah, so I have a couple notes too about like the general approach for this class. So the first note I have here on our like approach is distillation. And what I mean by that is that especially if you're a beginner, biology can really seem like a huge, intimidating field. It's just like a huge body of knowledge. If you're just a beginner and you like open up a biology textbook, it's like there's so much information there, so much knowledge, that it can be pretty intimidating, and it can be like difficult to know where to start. But with bioinformatics, we can get started doing cool and interesting analyses with only needing a couple of foundational concepts. Like we really just need like a basic foundation of some like pretty basic concepts to get started doing some of these analyses. And these foundational concepts will apply to literally every living thing, from bacteria to plants to mice to humans. The foundational biology we're going to be talking about is going to apply to literally every living thing. So it's about as general as you can get. And once we have this like basic foundation, that will be enough to get started with some of these bioinformatics analyses. And then from there, if you decide you want to go further beyond this course, that will provide you with like a jumping off point to then go further on whatever specific topic you're interested in. So like a good approach is to learn these like foundational concepts that apply across all of biology to every living thing, and then get a good grasp of the foundations, and then from there, whatever specific topic you're interested in, focus on like deep diving on that specific topic. So yeah, I just labeled that like general approach as distillation. So the next note about our approach is abstraction. And what I mean by this is that this course is meant for people who have a little bit of a background in programming and like hopefully like a little bit of data analysis and like working with data sets in Python. But we won't be assuming any biology knowledge. We're going to learn the biology we need, again, focusing on these like foundational concepts. And then we're going to try to abstract the biology into a data analysis problem that looks familiar to us. So basically, we're gonna turn this into a standard data analysis problem, and we're gonna abstract the biology so that we'll be able to actually open up the biology in Microsoft Excel and take a look at it in like a spreadsheet format that seems like familiar to us as programmers and data analysts. So yeah, that's kind of like the general approach is trying to turn this into a data analysis problem that will seem familiar to us as programmers and that we'll be able to get a hold of. Okay, so just to give you guys like a general overview too of the course, yeah, we're going to start with some of the biology, again, focusing on these like foundational concepts. And then during the course, we're going to do 2 data analysis projects. The first one is going to be something called differential expression analysis. That's a type of transcriptomics project. If you guys don't know what that word means, don't worry, because we'll explain that soon. And this is going to be using a real data set that I'm going to show you guys how to download from a government data repository that's publicly available. So yeah, we're gonna do a realistic analysis on a real data set from a real experiment, and they're going to walk you guys through how to do that in Python. And then the second project is going to be something called a GWAS, which stands for genome-wide association study. That's a type of genomic analysis. Again, if you guys don't know what this word genomics means, don't worry, because we'll cover it soon. This is going to be with a data set that's fake, but somewhat realistic. The reason it's with fake data is because usually these types of data sets, the genomic data sets, aren't publicly available due to privacy concerns about people not wanting their like DNA information publicly available. So this is not using real data because it's hard to find publicly available like real data sets for this, but it's going to be using a fake data set that I made up that is somewhat realistic based on like real data sets I've worked with at my job. I made up a fake data set that's just fake data, but somewhat of a realistic format to give you guys an idea of like what kind of information a real data set would be giving you and like how to work with it. And yeah, so like I kind of mentioned before, the prerequisite for this course is basic Python, including NumPy. So yeah, I'm hoping you guys know a little bit of like basic Python already. If you don't already know Python, I'll put a link in the description to a good course where you can learn some basic Python. But yeah, going to be assuming a little bit of programming knowledge, but no biology background knowledge is required at all. We'll start from scratch and cover everything we need to know in terms of the biology. So yeah, this is assuming a little bit of a programming background in Python, but no biology knowledge. This is for total beginners when it comes to biology, and we'll cover all of the necessary biology info. And yeah, so like the general goal is to use Python to explore these data sets and do some like basic analyses to look for interesting patterns. And in some ways, and I'll just say this up front, just to be upfront about what this course is going to be like. In some ways, the programming we're going to be doing is going to be slightly unrealistic in the sense that it's not going to be the type of pipeline that would be done in real life. Because in real life, if you're doing one of these analyses, the way people usually do them and the way that I do them, like at my job, for example, is just to use some software package, either usually in R or like just using like a command line, like terminal command line tool, and using like a software package that basically does most of the analysis for you automatically. So in real life, that's usually how they're done, just with like a software package in R that just does it all for you, or using a command line tool that just like does all of it for you. So that's how it's done in real life, but I actually don't think that's a good way to learn, because in some ways it actually makes it too easy. Yeah, like the thing to focus on learning is not how to like memorize a couple lines of R to just get it to do everything for you. That's not going to be the point of this course. The point of this course is to do it in Python as much from scratch as possible, so that we actually understand what we're doing and like actually understand what's happening every step of the way. There's going to be more of like a fundamental approach of learning what these data sets are, what is being measured, what information they're giving us, and how to work with that information to try to figure out something about the biology, and doing all of this ourselves, and like understanding what's happening at like every step. So yeah, that's the goal for this course basically. Okay, so before we start writing any code, I'm gonna actually spend the next couple videos telling you guys about the biology. So before we write any code, yeah, we want to build up this like foundational knowledge of the relevant biology. And yeah, but before we get too deep into it, I'm gonna just end this video by telling you guys the important part first. And then we're gonna expand more on this throughout the next couple videos and really like build up the knowledge we need to get started with these projects. But yeah, I'm just gonna end this video by like giving you guys a little hint of this important part first before we go more in depth on it. Okay, so this is the important part. So this slide is showing something that happens in every form of cellular life on Earth. So what I'm about to tell you guys is true for everything from bacteria to humans to plants to mice to fungi. Every type of cellular organism on Earth has this in common, basically. So all of these things have a genetic code called DNA in their cells. And the DNA has stretches of it, like segments of it, called genes. And these genes contain the instructions to make something called messenger RNA. And this messenger RNA is used to put together things called proteins. And then the proteins basically do like a lot of the actual work of the cell. So they're involved in things like signaling, stimulus response, metabolism, regulating various like cellular processes, making up like the structural components of the cell. Yeah, they basically do like the actual work of the cell. And so this process basically allows information that's stored in the DNA to like manifest itself through this intermediate step of mRNA and ultimately like result in proteins that are put together using this information that's stored in the gene. Put together these proteins that do the actual like work and functions of the cell basically. And yeah, just a little bit of terminology here. So the step where mRNA is made using the information in the gene is called transcription. And the step where the protein is made using the information in the mRNA is called translation. And you might hear the strands of mRNA sometimes referred to as transcripts. And yeah, so just to repeat myself, this is true of every cellular organism on Earth. And the cool thing about modern biology research is that it's possible to measure each one of these three things through like modern experiments. These things can all be measured in a way that we can get pretty interesting like data sets out of them. And yeah, pretty large data sets where as programmers our like programming and data analysis skills can really come in handy for analyzing these big data sets. So if you guys ever hear this suffix omics, so like for example transcriptomics, genomics, proteomics, the omics here basically means like the big data study of something. So there's a subfields of bioinformatics that focus on each one of these three things. And the omics suffix just means like the big data study of one of these things. So proteomics is the big data study of protein data. Transcriptomics is the big data study of transcripts, which again is just another word for these mRNA strands, and that will be the focus of our first project, transcriptomics, study of mRNA abundance data, basically. And then genomics is the big data study of genes or of DNA sequence data, and that will be the topic of our second project. There'll be a genomics project. Okay, so I think that's it for this video. Yeah, if that sounds interesting to you guys, I hope you'll stick through this course and continue watching these videos. Yeah, we're gonna be going more in depth on all of this stuff, and yeah, should be pretty cool and pretty fun, I hope. Yeah, so thanks for watching, and see you guys next time.